Hello, everyone. I'm James Peterson. I'm the Senior Director of Operations and Outreach for the Center for Physical Genomics and Engineering. And I'm here to introduce the speakers for today for the Physical Genomics Training Program, or the PGTP Research Showcase. So the PGTP is a NIGMS funded T32 training program with the mission to train the next generation of transdisciplinary scientists that will bridge molecular biology, bioengineering, physics, optics, chemistry, and medicine. For those that are kind of new to physical genomics, sort of broad descriptions, a new field that aims to understand from the first principles of physics, the structure and function of the genome and the role in generating global pattern of gene expression. So the three-dimension structure of chromatin and how that sort of affects the global patterns of gene expression. So Dr. Backman, the director of the center, as well as the training program, likes to use the analogy that if DNA is the hardware and epigenetics is the software, the three-dimensional structure of chromatin is the operating system, which then enables global gene expression and ultimately cell phenotype. So, like I said, Dr. Backman is the director of the center and the director of the program. Unfortunately, he is on bereavement leave and he was not able to join us here today. I am James Peterson and the senior director of the outreach and operations for the center. And then Ben King helps administer the program as well. Program comprises 42 faculty from 3 different schools and 17 different departments. We have currently 16 trainees part of the program, and we'd like to welcome Maddie Massengarb as well as Jiang Yang, 2 of the newest trainees in our program that just started this month. So getting to the research talks today, we'll hear from Karla Medina. Advised by Dr. Backman and Dr. Matei. She'll be presenting on investigating the role of three-dimensional chromatin domain organization in ovarian cancer stem cells. Dylan Howe was slated to present, but unfortunately he got COVID. So he is unable to join and is not feeling well to present. But we'll get him next time. And then Esther Yoon from Gemma Carvill's lab, and she is co-mentored by Shiji. She'll be presenting on investigating the role of lncRNA Chaserr on CHD regulation and neurodevelopment. And so with that, I will go ahead and turn it over to Karla. And you can go use my computer and everyone should be able to hear you.
Perfect. Hi, everyone. My name is Karla Medina, as James introduced me. So I am now a 4th year in the Backman Lab, and I specifically do a lot of nanoscale microscopy, which I'll share with you to understand how that organization of chromatin facilitates the phenotype and function of ovarian cancer stem cells. So I'll give you guys an introduction to the three-dimensional structure, why it's important to look at the physical organization and what that means, and how we can actually visualize this using different nanoscale imaging microscopy methods. So we have a platform called NanoChia, or Nanoscale Chromatin Imaging Analysis platform, and I'm using this to primarily, first and foremost, assess the bivalency in cancer stem cells. So without further ado, so the physical organization of our genome, if we were to stretch out our DNA linearly, it'd be about 2 meters in length, right? We have 3 billion base pairs of DNA, but yet that's somehow compacted in the 10 micron size nuclei. How that compaction is actually organized, and because in the field we've been able to debunk that the 30 nanometer fiber is ceased to exist without experimental manipulation that would cause us to see that 30 nanometer fiber. We understand that we go from our DNA to beads on a string, but we still are, the field is still working to define what is that intermediate structure before we go on to fold into mitotic chromosomes. So that's where much of my work persists. And us in the field, as well as others, have looked at how this organization, so changes, and specifically in the interphase stage of the cell cycle. So that's the time that the cell is spending majority of its time in, because that's where most transcription is occurring. So understanding how that organization and that structure forms during interphase is widely important because it relates to all that bulk RNA sequencing outputs that you get in return. So what you et al. In the research group found was that actually chromatin can be described as a distorted polymer chain, taking on lengths between 5 and 24 nanometers. Now, working in my lab, we do a lot of ChromStem, or chromatin scanning transmission electron microscopy. And what we do is take ChromStem images. So this is an image of chromatin density. We can visualize high versus low density regions because our technique allows us to do direct DNA labeling. So anything that we see as signal is a direct measurement of the density or the amount of DNA that's there. So we can kind of separate high versus low regions. And then we can apply a local maxima technique that will allow us to parse out and visualize those high density regions, and then calculate the centroids of those regions that are found and define those as our domain centers. And what we see when we measure out the packing scaling or the mass scaling properties as we move from that center point outward is this mass scaling behavior. So meaning that the amount of mass that's occupied within one of those domain centroids is actually correlated to the amount of the space that it's taking and to the mass scaling coefficient, which we label as D. So this is our statistical metric for understanding how that chromatin compaction is actually being exhibited within the space that it's occupying. And then again, when we measure radial density from the of the centroid outwards to the periphery, we see that it's very, very dense, and then you see a drop off, but then you also see it picking back up again. I don't know if you can see my pointer. So the point at which it picks back up lets us also know that these, yes, are separate domains, but they are also interconnected by linker DNA. And when we did this across A549 or lung cancer cells, we saw that this takes on a radius size roughly of about 80 nanometers. And so altogether, we can use ChromStem to analyze those structures of packing domains. And here we have the example of the A549 lung cancer cells and see how that changes between a healthy cell like a human fibroblast cell. So this is an important distinguishment, and I'm utilizing this feature to understand how those packing domains are then organized in ovarian cancer stem cells. So ovarian cancer is a solid base tumor that begins within the epithelial lining of the ovary. So in the ovary, upon accruing aberrant mutations, what we see is the development of these tumors or these cancer spheroids, which within this population, there's a smaller subpopulation of CSCs or cancer stem cells that can actually regrow and repopulate new tumors. So cancer keeps coming back, and CSCs are predicted to being the underlying culprit because they allow for tumor regeneration because they have a lot of multipotency capacity that allows them to differentiate and formulate these tumors. In addition, they're also very highly chemoresistant. So after chemotherapy, you might see initial progression. So majority of patients upon first treatment, 70% of them will respond well, but it's within the 5-year time point after chemotherapy treatment, we see a lot of relapse. And specifically what we're noticing is between these CSCs or these cancer stem cells, so essentially cells that have stem-like properties, cancer cells that have stem-like properties versus the non-CSC, which is like a baseline tumor cell, we see also differences between their chromatin markings or the epigenome features between these two cell types. So we can actually distinguish them, or the field is working towards distinguishing them by these features. For example, what we see on the right is that the ZEB1 gene, which is a facilitator or regulator of EMT processes or endothelial to mesenchymal transitions. This is actually found in breast cancer. So again, CSCs are now widely identified in a lot of solid base tumors. originally founded in AML or amyloid leukemia, myeloid leukemia. So the ZEB1 gene is left poised in the CSC state, but it's left poised, meaning that it's ready and it's primed in order to turn on gene expression at the cue of like a signal. So like any extracellular signal, like a cue in inflammation, could trigger the CSCs to start regrowing. Otherwise, they're left heterochromatically repressed in the non-CSC state. And then we see this is true at multipotency gene sites where we're leaving these genes on and open and accessible, whereas in the non-CSCs, we have them heterochromatically expressed and then repressed. And then in, again, the tumor suppressor genes, we're leaving these off so we can allow tumor growth to occur. And otherwise in the non-CSC, they are left on. So we see they have a higher potential for formulating these new tumors, and we want to be able, or I want to be able to utilize our nanoscale imaging platform to be able to distinguish these features down to that nanoscale level. So those changes in chromatin organization, we know we can distinguish OCSCs apart from non-OCSCs, and the baseline criteria for ovarian cancer CSCs specifically, because they vary depending on which cancer type you're referring to, glioblastoma, pancreatic, colon, or breast. So for OCSCs, we denote them by their surface expression marker of CD133. They also have high endogenous expression of aldehyde dehydrogenase, which is our marker for distinguishing them. So, and actually both of these features, CD133 and ALDH, are used widely in the field to distinguish between stem cell and non-stem cells. So aside from cancer stem cells, but now being applied here. And then as well, showcasing that self-renewal capacity and sphere forming capacity. So this showcases that, yes, they are in fact able to regrow out new tumors because they showcase sphere forming or tumor initiating properties, whether they're in vivo or in vitro. And then again, the Yamanaka factors with SOX2, OCT4, and NANO. So a goal in my lab is to actually see and how we can relate chromatin packing scaling as a biomarker to distinguish these CSC populations, and then further dive into how those packing domains are actually organized. What kind of histone markers do we find at their core versus their peripheries, and the size of their packing domains as well. So we first and foremost, I utilize PWS, which is our lab's partial wave spectroscopic microscopy method. It captures an image cube of data because it's capturing a series of images across the entire wavelength of light. So at every 2 nanometer increment from 400 to 700 nanometer wavelengths, we're capturing the backscattered reflected light that's coming from the back cell or the glass cell interface. And then after capturing this, we're using this to distinguish between, is there a difference in the chromatin packing scaling? Because we can take this on the average of a single nuclei, but then also take that average across other cells within that same population. So it kind of gives us a more high throughput analysis of what that packing scaling looks like globally within the nuclei. So we take our average nuclear D or chromatin packing scaling measurements, and we saw that in the ALDH positive cells, so our CFC cells, we have a roughly about a 5.6% increase in packing scaling compared to the non-CSC group. In addition to PWS, I will also be employing STORM and ChromStem. So STORM is a super resolution technique. So it stands for stochastic optical reconstruction microscopy. And what it allows us to do is actually get 12 to 20 nanometer localization precision. AKA, so comparing this technique with your standard immunofluorescence methods, what we're doing is roughly the same. So same sample prep, we're tagging our primary antibody to our secondary fluorophore, but instead of shining it with fluorescence lights, we're actually using lasers. So that way we can allow these fluorophores to essentially switch on and off. So they go from an excited state to a ground state. And because of that, we're doing 10,000 frame capture. So we can localize every single point at which that fluorophore was found to be on or blinking. And then we can reconstruct this image using a thunderstorm, a plugin package on image J and get our super resolution reconstructed image of all the localized fluorophores that were found. And then with ChromeStem, we can identify those packing domain features. So now kind of narrowing back into what do each of those individual packing domains look like, how are they organized, etc. So first step, as I mentioned, I'm trying to do this to understand bivalency. So apologize there for the mishap, but essentially the markers that I'm looking at are H3 or histone 3 lysine 27 acetylation, which is a marker of active enhancer elements, H3K4, which is a marker of active gene promoters, and then H3K27, which is largely found or widely found across developmental gene and chromatin regions. So again, bivalent states, meaning that we're having two markers present at the same gene site roughly. So what we've taken from some early ChIP-seq analysis is actually understanding, and this is just hyper-focusing onto one single gene, the ALDH1A3 gene, where we see evaluating the differences in peaks between the ALDH negative and the ALDH positive cells. We see that there is actually this kind of characteristics where the CSEs are being primed with H3K27 acetylation and K4 trimethylation, where non-CSEs are primarily marked with K27 trimethylation and K4 trimethylation. So my goal was to then understand how this is actually relating, because ChIP-seq is a one-dimensional analysis, and that's kind of what we employed here. So taking it linearly from a chromosome to chromosome state, we can see that the relative levels of K4 expression, or that concentration, is directly correlated with the amount of K27 trimethylation expression that you would see. So wherever you see a K4, you're more than likely to also see a K27 marker. Then we did this analysis too with K4 and K27 acetylation and K27 trimethyl and acetylation markers together. And we see a pretty strong correlation between the two marks. That tells us that the CSEs actually do in fact showcase a lot of bivalency that's not necessarily witnessed in the negative cells. So my approach, so I did some storm imaging, and I wanted to see, because again, at the baseline, bivalency means that there's two marks at the same point or positioned on the same gene. So my approach in thinking through about this, that I employ first a nearest neighbors model, essentially finding all the K27 trimethyl points that are within another K27 acetylation point. So trying to see where in the spatial proximity, where are the 2 points nearest to each other, and I gave it a threshold of 20 nanometers. Then I applied a CKDT tree to then find those intersections between the K27 trimethyl points, all those that were identified and grouped together. So that way I can see, okay, this was a true, like it's not just an artifact, it's a true expression of K27. And then I plotted those to see how close they are to the nearest K27 markers that were then found. And so doing storm imaging, I think this is what makes the work very exciting, is because you can get these really beautiful images of what is actually, what the cell actually looks like in terms of this expression. So it kind of gives more of that like realistic feature as opposed to sequencing, but I don't hate on sequencing. It's very valuable, but now I'm just trying to see if what we see in 1D is also true in 3D or 2D in this matter. So applying this technique, I scrutinized to, again, find all those nearest neighbors between each group. So I did this between the K27 acetylation group. I don't know why this has been covering. No?
You gotta use the other one.
This one.
You gotta use the other mouse for the computer. Sorry.
Oh,
Ugh, sorry.
sorry. Just so you guys can see for reference. So I applied my nearest neighbors within 20 nanometers to identify all of those neighbors in my K27 marker expression, and then K27 trimethyl marker expression. And then I created a merged image, kind of, so to speak, which is only visualizing those points that are intersecting, because my bottom line question was, wherever I see a K27 trimethylation mark, do I also see a K27 acetylation mark? Do they overlap? Do they intersect? And thus, if they do, then I would be able to distinguish those as being bivalent. So, sorry, before I wrap up on this point, this is a very crude analysis approach, so I'm also very open to feedback. But in this aspect, wanting to just see, does bivalency exist between my two groups? And although it's very tiny to see here, I did get about roughly 82,000 points of intersection in my LDH positive group versus, I think it was 55,000 in my negative group. So I do see a difference between them, and you can kind of see that qualitatively here. So next, I mentioned about ChromeStem. So just to give you a brief background on this technique. So I apply this in both 2D monolayer cells and 3-dimensional spheroids. So as I briefly mentioned, it's a direct DNA labeling technique. So that's essentially what I'm doing here. I fix the cells after growing them, plating them, and allowing them to grow overnight. Then the next day, I'll start my ChromeStem processing, which I basically do a chemical fixation, but then I also do my DNA labeling. So I use DAB and DRAC5. So DRAC5 binds directly to the major and minor group backbones of the DNA structure. DAB polymerizes upon the excitation of light. So we use 680 nanometers or a Sci5 filter to allow this polymerization to occur and essentially enhance that contrast. So we photobleach the cells so that way we can mark the DNA. And then we further enhance that contrast using osmium staining. So then we can apply a heavy metal dye staining to further enhance it. So when we take it under the microscope, we can easily distinguish between those high and low or high density versus low density regions. We then embed them into resin, section them, and then image with our STEM microscope that's over at the nuance core. And what we do is do a 3D capture. So as I shared those 3D renderings of the voxels that were taken between the lung cancer cells versus the fibroblast cells. How we do that is we're taking, again, like 2-degree or capturing from the positive 60 to negative 60 axes at every 2-degree increment. So we get in total about 121 images. We apply gold nanoparticles to our section so that way we can do a reconstruction and then get those beautiful images that we see. The process is fairly similar to spheroids, except it gets a little bit trickier, and I had been working majority of this year trying to optimize this protocol because these are spheroids. They grow on ultra low non-attachment plates. So I have to embed them into agarose so that way they can kind of essentially stay still while I do my DNA labeling and then process them for the remaining steps of our Chromium protocol. So that's currently what I'm working on right now. So I'm taking my LDH positive versus LDH negative spheroids. This is after 1 week of growth. And I mean, it makes it a little bit challenging because in my LDH positive group, I get these defined spheroid structures, which here, not to confuse, it's not this whole structure, but it's all of these inner spheroids that you see here. So in comparison to the negative group, which just doesn't make as many spheroids as the LDH positive cells. And all I do is essentially fax sort them by this marker and then plate them and then feed them every 3 to 4 days. And so currently the step that I'm working on is optimizing that DAB polymerization step. So that way I can do all that DNA labeling and then enhance it and then be able to place them into resin section and then image with the ChromeStem microscope. So one of the things that we're playing around with doing is we do this polymerization between 2 to 7 minutes. And I'm also moving between the Z planes so that way I can effectively excite or allow depolymerization to happen from the bottom of the spheroid to the top. And so, yeah, sorry, that was just to recap, that was all for today. More or less progress research updates. I can't make any conclusions off of this work yet, but I plan to in the near future. And I just wanted to take a moment to all my amazing lab mates that helped me devise this this research plan and proposal, and then also carry it out. So I got a lot of help from everybody, and I'm very grateful. Yeah, and we'll open the floor for any questions.
Questions? There's anything in the chat. Yeah, go ahead.
I had one question about the analysis you did with the storm images. If you can go back to the slide. Yeah, so here you said that, just to like clarify, do you identify the intent, like the peaks of the histone markers labels, and then from there you draw like a 20 nanometer radius? And then whatever, if there's like another peak within that radius, even if it's just like a single peak, do you label it as they're like near each other? Is that how the process works?
Kind of. It's not exactly like peaks, but just points.
Yeah.
Because each is a localized point. So I'm just trying to see within a 20 nanometer radius of one point of K27, for example, I find the next nearest point that's within that 20 nanometer radius. And then I call that like a nearest neighbor.
So is all the intensity that is on the left image, all the peaks that are like
Yes,
within,
essentially.
okay.
And then I gave it a much lower threshold when I find the intersecting, because for intersecting, I'm saying that first, find me all the high dense regions of K27, find me all the high dense regions of K27 acetylation. And then now to distinguish between, are these two in overlap within the same coordinates, I give it a 11 nanometer threshold. And I picked that just very roughly because of the size of a nucleosome. So essentially, if they're going to be found spatially together or potentially on the same gene, then they should be in a very close proximity. Yeah. But I did the first two steps to kind of filter out the noise and see it from there.
Yeah, I guess like for this, are you concerned about the uncertainty limit of like storm? I know we usually get around, depending on like the labeling, but like 10 or the 12 to like 20. So I just want to know your thoughts on like how you're going to address that moving forward.
Yeah, I mean, for doing the nearest neighbor search, I also try to eliminate any points that are considered noise. So if it's not within this bound or this radius boundary, then it's considered a point of noise. So in order for it to be a highly expressing region of this particular histone marker, it should be within another marker there as well. I don't know if that makes sense. But yeah, I tried to eliminate the noise, but I think one of the next questions like I want to get to is to identify like packing domain organizations specifically. So our lab has proposed differences between like what histone markers you'd likely find in the inside of a core versus the outside of or peripheral region. So we denote packing domains being organized potentially with a very dense heterochromatic core with more H3K9, which is a known heterochromatic marker. And then at the peripheries, more euchromatic markings such as K4 and K27. However, when I image with K9, and why I don't have it here is because it's largely found around the periphery of the nuclei and not necessarily, so it's more like, yeah, like, yeah, around the periphery of the nuclei, not exactly in the core where it's likely being used to control gene expression. So I'm working on my hypothesis now with testing how K4 and K27 cores. So I have that analysis, but I don't have it really fleshed out well. So I didn't show those images, but did the same labeling to identify, do CSCs form more K27 domain core regions or K4 regions? Especially if K9 is at the peripheries, then likely there's something else that's driving that domain organization in the innards of the cell.
Did you see Ollie's question? He asked for why did you choose a threshold of 20 nanometers for your nearest neighbor-based classification approach?
So I did increments from 4 nanometers to 6 to 10 and then to 20. I didn't go beyond because I'm not trying to find so I tested out at various different thresholds, and it seemed like 20 was the most representable for the data that I was working with, or the data that I'm working with, because essentially I'm not trying to find, so an alternative to this was doing dbScan clustering, but in caveat to that, because my question is more or less, is there bivalency? So I wanted to distinguish high expressing regions of K27 versus low expressing regions. So given the size of a packing domain is roughly about 80 nanometers, as we described, I didn't want to go anywhere beyond that or close to that, because my prediction is also that CSCs are forming a lot smaller packing domains, chromatin domains. So although I don't have those values now, it just seemed like this was the most representable for now, but that's not really like a tried and true like threshold. It still is just like a crude estimate of like what is the most optimal. So yeah, there's a bit of bias, but I just tried to stick within a threshold that was going to be able to tell me, okay, where spatially do I see the highest amount of K27, points or peaks.
Yeah, there's another question.
ALDH is a marker for the defined by the field. So as I mentioned, so there's been studies that have done this excessively across all different types of CSC populations that have been identified. Because again, CSCs have been widely described across a lot of other solid-based tumors, including the AML of more like blood-based cancer, where we see there's differences in these markers, but CD133 and ALDH are both markers of stem cells and highly associated with ovarian cancer stem cells specifically, as opposed to some other markers that you might see like CD45, for example, that's widely used in breast cancer. CSCs.
So I was just wondering what the role of ALDH is in bivalency control. Wouldn't you want to, if you're looking at bivalency, wouldn't you want to look at something also that is a known regulator of bivalency genome-wide?
That's a good point. I don't think that we know that though yet.
There are plenty of factors. The 7SK is an RNA that'll regulate genome-wide bivalency. There are a lot of factors that you could look at that directly regulate bivalency genome-wide, or I don't know about directly, but will, change bivalency of many, many, many targets, right? So that's a known thing. So if you want to study this, I think that would be a good to include in your studies. Not that you shouldn't use ALDH, it's fine for your cancer question. Well, I don't know if is ALDH mutated in ovarian cancer. Is it a known direct link to that?
Mm,
It's just a marker, but is it a functional gene in ovarian cancer?
Yeah, so LDH is more related to the metabolomics of the cell. So it regulates or combats against negative or harmful aldehydes. So I'm using it here just to distinguish, so from a homogeneous population, because I work with a commercially available ovarian cancer cell line, but yet I can grow them out, get a high number of them, take them over to the fax machine, and I use aldefluor kit from Stem Cell Technologies to distinguish between high versus low LDH expressing cells. But that's more of a stem cell feature, not a bivalency regulator. So I think,
Oh,
or
okay.
I guess like what I was thinking about was maybe, so like ZEB1, for example, it's highly known for regulating that bivalency because it's a regulator of EMT, and it's also associated with regulating the interconversion between non-CSCs and CSCs. But you raise up a good point. So I would presume potentially as if there are surfers markers for it, but I guess the point here it was just to distinguish between what's a CSC versus what's not a CSC. But then looking into global regulators of bivalency would be maybe used to just enhance that, maybe like by
Well, I think it would help validate your methods because you're using a bunch
Yeah.
of new methods, right? So that's what you would use it for, not necessarily, I mean, it would be relevant to the stem cell question, of course, because, bivalency is a marker of boys and not boys genes. So that's always a good thing to look at for your stem cell question. But I think that because you're trying to develop these new methods, you could use that. Could use looking at something that's known to regulate bivalency genome-wide, and that way you could see how well your methods are correlating with that expectation. You know what I mean?
Yeah, no, thank you. Yeah, I'm noting that down. Appreciate that.
Right. I think that was great. Way to go. Thank you very much. Next, we'll move on to Esther's presentation. The mouse will show.
Oh. This one. All right, great. Hi, everyone. I'm Esther. I'm an MD PhD student in Gemma Carvill's lab. I'm a third year in her lab as a PhD student. And today I'd like to talk to you about investigating the role of long non-coding RNA chaser on CHD2 regulation and neurodevelopment. So CHD2, the gene encodes for chromodomain helicase DNA binding protein 2, which is a chromatin remodeler. And it's kind of a nice segue with Karla's because she talked about bivalency. And here I have a picture of a poise promoter. So in this case, how CHD2 functions is that it interacts with different transcription factors and cofactors and binds to such poise promoters. And when it does this, it deposits histone variant H3.3, which allows for a more open chromatin conformation. And this allows for Pol2 to bind and promote target gene expression. And in mice, when CHD2 is knocked out, there's an increase in the repressive histone modification, H3K27 trimethyl, and this leads to premature neuronal differentiation in mice. And importantly, in humans, de novo loss of function variants in CHD2 lead to neurodevelopmental disease. So when I say loss of function variants, this is like truncations or deletions or missense variants. And in humans, when you have only one functional copy of CHD2 due to these loss of function variants, you have a neurodevelopmental disease consisting of seizures, wide range of motor, intellectual, and developmental delay. And because of
you only have one functional copy, and that's not enough to promote healthy neurodevelopment. The mechanism of disease is known to be haploinsufficiency. And now I'm going to switch gears to talk about Chaserr, which is a long non-coding RNA, here shown in purple, upstream of CHD2. And so far, we have identified 3 individuals across the world who have a deletion of Chaserr. As you can see here in red, while the deletions are all different in size, they all are deleting the 5' UTR region as well as the first 3 exons of Chaserr. And what is interesting about these deletions is that it seems that it is due to a ALU-ALU mediated non-homologous allelic end joining. And what I mean by that is that there are a lot of elements called ALUs, repeating elements in the genome, and they have this microhomology so that when that they spliced out the Chaserr region because it mistakenly joined together. And you can see the breakpoint junction microhomology for all these 3 different individuals. And something that is also more interesting to me, as I'm interested in being a clinician, is that Chaserr deletion actually causes a more severe disease than CHD2 haploinsufficiency. So these are the 3 identified individuals so far, and you can see that they have some shared facial dysmorphisms, such as wide-set eyes, upturned nose, low-set ears, and a long filter. Whereas in the CHD2 haploinsufficient individuals I've met through family foundation meetings, they all seem to look more like their siblings than to each other. And also, while the CHD2 haploinsufficient individuals, they have seizures, but a lot of them are able to go to school and like play sports and even hold a job and do things independently. These individuals, the Chaserr deletion patients, they are wheelchair bound, like extremely hypotonic, so they can't stand or do anything. They're not verbal. So it really seems like a more severe disease. And also you can see here the 3 different MRIs over time at 1 month to 4 years. And you can see predominant prefrontal cortical atrophy, as well as reduced volume of the brainstem and hypomyelination of the corpus callosum. And where in contrast with the CHD2 haploinsufficient individuals, they have a normal brain MRI unless they have very severe treatment-resistant epilepsy and seizures that lead to epileptic encephalopathy. So seems like Chaserr deletion is more severe, and how are the 2 related? And so in our lab, we got fibroblasts, punch fibroblasts from the 2 different individuals with the Chaserr deletion and reprogrammed them into iPSCs. And when I performed Western blots to look at CHD2 abundance in these 2 individuals as compared to wild type lines, as well as a CHD2 knockout line, you can see the darker band in the 2 different Chaserr deletion individuals, showing that there is more CHD2 compared to the loading control. And when I quantified this, it shows nicely that in the individual 1 and 2 with the Chaserr deletion, compared to sex-matched wild-type controls, they have an increase of CHD2 abundance of about 1.75-fold. And then to see if there's this, if this is in cis, what we did is we performed long-read sequencing on the Chaserr patient lines, and we phased out which CHD2 allele is in cis with the Chaserr deletion using these SNVs that are in their genome. And then once we phased those alleles out, we performed RNA-seq and high-depth sequencing. And from that, we found that compared to GTEx wild-type controls, in the 2 different Chaserr individual lines in different tissue types, you can see that in blue, the CHD allele that is in cis with the Chaserr deletion is overexpressed compared to the 50-50 balance you would see in the GTEx controls. And so with that knowledge, my 2 questions really focused, one on the mechanism, how does Chaserr regulate CHD2? And second, what are the downstream consequences of too much CHD2, as previously in our lab, we studied a lot about CHD2 having too little CHD2 and how that is bad for neurodevelopment, but it actually seems like also having too much CHD2 is also not great for proper neurodevelopment, and you need to have a very tight regulation of CHD2 gene dosage. So in order to, before I dive into the mechanism, I want to introduce you to Exist, which is a lncRNA that is a well-known model on so far on how lncRNAs can be used to regulate gene expression. So far, or historically, long non-coding RNAs have like not been super well understood because they don't actually, like their name, they're long and they're non-coding, so they don't have a functional protein output. So they thought, oh, it's not very important, but now we're having more understanding that they serve regulatory roles, and one of them is being Exist. And so this, the lab at Caltech called the Backman Lab, they developed this protocol called CLAP, covalent linkage affinity purification, where they covalently tagged a protein, in this case SPEN, and they did cross-linking and used stringent wash and denaturing conditions to pull down any RNA that is bound to SPEN. And they found that SPEN, a repressive protein, binds to Exist, specifically at the A repeat region in exon 1. And furthermore, they found that when long non-coding RNAs are transcribed, they're spatially concentrated where it's transcribed, and it helps to recruit the SPEN protein, which then amplifies and recruits more of the SPEN protein itself, and then helps to silence the X chromosome at a chromosome-wide level. And so last year, I was at the Regulatory RNAs Keystone Conference, and I talked to Prashant, who is one of the MD PhD students in the Backman Lab, and he helped develop this method called RD Sprite, where they imaged RNA and DNA in the spatial, in 3D spatial resolution. And what they found for Chaserr is that the RNA is really localized to the CHD2 Chaserr area. And this is a different view from his paper, which shows that you can see here, there's an RNA called MALAT1 where it's kind of everywhere. So you can see that through the different, kind of the whole chromosome, whereas in Chaserr, it is quite localized to its locus in chromosome 7 in mice. And then Exist is you can see that it is kind of all over the X chromosome, but not anywhere else. And then what he shared with me is that they use the same technique, CLAP, and they found that SPEN also binds to exon 5 of Chaserr lncRNA. And then he sent me over some of the lines that their lab has generated. And so this is a line that they made where there is an endogenous SPEN that is tagged with an auxin-inducible degron and a halo tag. And so when you introduce auxin into the culture, it tags this line with E3 ligase, and it becomes ubiquitinated and degraded. And then furthermore, they introduced a DOX-inducible piggyback construct of the full-length SPEN, as well as halo, so that when you have DOX in the media, there's exogenous SPEN in the cell. So then I used this and performed a lot of different Western blots. So I'm just going to walk you through here. So these are the 4 different, just like wild type, no DOX, no auxin condition, where you can see the SPEN halo around 500 kilodaltons, CHD2 here, and TAF5 as a loading control. And you can see that when I add auxin, SPEN gets depleted, endogenous SPEN gets depleted. And then when I introduce exogenous SPEN with DOX, you can see it come back up. And when I quantify this using densitometry over like maybe 10 or more Western blots, you can see that compared to the wild type, when SPEN is degraded with auxin, you see an increase in CHD2 levels, showing that potentially SPEN is a really important repressor of CHD2 via Chaserr. And that when I reintroduce exogenous SPEN with DOX, it seems that the CHD2 protein levels seem to go back similar to the wild type levels. And then when I overexpress SPEN with both endogenous and exogenous SPEN, it seems like CHD2 protein levels actually get even more repressed compared to wild type. And so my current working model is that Chaserr recruits SPEN at its locus where it's concentrated to modulate local chromatin architecture and control CHD2 expression. But this isn't the whole story. I need to figure out other players in the Chaserr SPEN interaction complex that helps to actually repress CHD2. So right now I am doing cut and run. It is a very similar approach to ChIP-seq, but it's really good for looking at chromatin remodelers. I'm happy to explain more if you guys have questions on it. But basically, I have a CRISPR-Cas9 knockout line of Chaserr that I made, and using wild type and Chaserr knockout hap1 lines, I'm gonna look into where SPEN, CHD2, and a lot of different histone modifications are in the genome, especially at this locus. So I don't want to spend too much time on it, but some of the things I'm interested in is HDAC3, which is known to be recruited by SPEN in silencing the X chromosome. So it's a histone deacetylase that deacetylates H3K27 acetyl. So I'm interested in looking at that at the CHD2 locus. And then also this approach cut and run can be used not only to look at what is happening at the local level, but also to examine what is too much CHD2 doing in the globally in the genome. So, and with that, I think my project can contribute to, what is too much and too little CHD2 doing, and then combining that with RNA-seq to study how is it affecting gene expression and regulation as well. And so with the PGTP, so the Carvill Lab isn't really an imaging lab, but I learned a lot with the PGTP and learned how cool imaging is, but I don't have the skills to really execute, but I think this would be really cool. Pictures speak 1,000 words, and I do have this line where SPEN is tagged by halo, and if I could just visualize with maybe CHD2 DNA, where CHD2 locus is, and then with the Chaserr RNA, where that RNA is, and then also tag SPEN with the halo tag and see if they all can be visualized in the same place. And I think this would really add to the story that it all works together. So if you want to help out, and I'm very happy to talk after this. And with that, I just would like to thank my lab, the PGTP, the MSTP, and then also Prashant and the Backman Lab at Caltech. And